So you may have heard of epigenetics as a way to control your genes and choose what you want your genes to do and take control of your own genetic destiny. There have been a lot of kind of snake oil pitches about epigenetics recently, and today we're going to kind of get into that and whether or not those are true. So I guess first I want to explain what is epigenetics?
So before we talk about epigenetics, it might be helpful to review a few genetic principles. So we're going to be talking a lot about this word phenotype. So when we talk about phenotypes, really what we're describing are different traits. Like I have brown hair, Emma has red hair. Phenotypes can be a little more complex than that, though, when you start talking about things like intelligence. So your phenotype obviously comes partially from your genetics and the DNA that you inherit from your parents, but also a huge driver of phenotype can be the environment in which you live. So it's not just your genetics that's defining these traits, but it's a mix of your environment and your genetics. Now, what that mix is that a 50-50% mix, that will probably depend on the trait. So for hair color that I mentioned before, that's more driven by genetics.
You'd think so, but I recently did 23andMe and they actually test the gene that causes red hair, and they told me I was only 6% likely to have red hair.
Wow, really?
Yeah, so
I did not know that.
Somehow my environment plays a huge role in me having red hair, even though people in my family have had red hair. So I was shocked at that, because I'm like, oh, hair color, especially for redheads, is determined by these specific mutations in this gene, but
so it's always a mix.
Not.
So now that we understand the concept of phenotype, what is epigenetics and how does that contribute to our various phenotypes?
Yeah, so the easiest way to think about epigenetics is to break down the word. So epi means above, and then genetics means genetics. So you can think of epigenetics as being above genetics. So say if you think in terms of a Word document and a paper you're writing, you can bold different words, you can underline different words, you can highlight different words, and you're not changing the words themselves, but you're just putting more emphasis on them. And epigenetics is really similar too. So you can have a gene and you are adding certain marks that make a gene more likely to turn on or a gene more likely to turn off. So one of these marks is called methylation, or the addition of a methyl group. So methyl groups are just this molecule of a carbon with hydrogen atoms attached to it. And methyl groups are found in foods such as strawberries, fish, eggs, and the more food you eat that has methyl groups, the more your methyl groups your body will have to methylate gene.
Epigenetics shouldn't be confused with actual mutations in your genes. So if we're going back to that analogy of the Word document, a mutation would be like a misspelled word. It's on print, it's in ink, you can't change it. Whereas epigenetics is more like track changes, it's dynamic.
Right, it's reversible.
Yeah.
So you can bold that word and you can unbold that word. So it makes, with epigenetics, a lot of things in your body can change based on what you're eating, exercise, environment. It's a lot more dependent on environment.
So why is it important to turn genes on or off? Wouldn't it be better to just have them all on?
There are certain genes, like what we call oncogenes, where they promote tumor formation. And those are examples of genes where you definitely want those genes to turn off. And there are other genes that may secrete molecules that tend to keep you awake. And at certain times of the day, you probably want those genes turned off.
So you mentioned methylation as one kind of epigenetics modification. Has there been any research on this topic?
Yeah, there actually has. There was a really cool study published, I think it was a few years ago. And what they did, they were trying to measure stress response and look at it epigenetically. So within our brains, we have what's called the Glucocorticoid Receptor, and this receptor is activated under stressful conditions and produces molecules that help you deal with stress. And what they did in this study is they used mice, and they had a group of mouse moms that would lick and groom their pups, and they had a group of mouse moms that would not lick and groom their pups. And this was a genetic change that was different between the mouse mothers. So they wanted to see how the pups fared under stress when they were licked and groomed by their mothers versus when they were not licked and groomed by their mothers. And one interesting thing with mice is that at the beginning of their lives, the glucocorticoid receptor gene, it's very highly methylated, so it's shut off and it's not active. And what they saw is that in the mothers who licked and groomed their mice, those pups that were licked and groomed, they actually had activation of the glucocorticoid receptor gene, which turned on the gene so that the mice could better deal with stress. And the way they kind of measured stress in these mice is they put them in what's called an open field test, and it's basically where you have an open area and you see whether the mice will walk around in the middle or the mice will kind of go on the side. It's kind of like your introverts and extroverts at a party. Your introverts will kind of stay towards the wall and not be in the middle of people, whereas your extroverts are wanting to be out around people. So they saw that the mice who were not licked and groomed would tend to stay towards the wall, and showing they were kind of highly stressed because their glucocorticoid receptor was not turned on and they were not able to deal with the stress properly. So this is really interesting in the context of methylation, because methylation is the thing that was changing in these mice based on whether their mothers licked or groomed them. But one thing to note is, if you think about mice, if they're in the wild, it's probably good for them to be more anxious and be more aware of their surroundings if they're trying to run away from a predator or find food.
You mentioned like a nurturing environment contributing to the methylation, but we also talked about how depending on your diet, you may have more methyl groups available. So, I mean, how does diet play into epigenetics?
Yeah, diet actually plays a lot larger role in epigenetics than you would think, and there's tons of research being done into this because it's kind of a newer field. So one way we can think about this is in terms of cancer. So in cancer, there are genome-wide changes that will happen to your genome because you have cancer. And specifically, especially in the cancer itself, the cancer will have a different genetic profile than the rest of your body will. And the genetic profile that scientists see in cancer is this global demethylation in cancer. So all the cancer cells will have like no methyl groups on them, which means that they're basically, all the genes are turned on and active. And this can lead to the cancer cells growing uncontrollably, the cancer not responding to treatment, the cancer being able to morph and change and get genetic mutations in places that would not normally be possible in a normal cell. So scientists have found that tumor suppressor genes, which are the genes that are active and are the first line of defense against tumors, that half of these tumor suppressor genes are inactivated epigenetically. So which means that they've had a methyl group added to them, or they've been mutated and they are not active in cancer. So your first line of defense is knocked down just by epigenetics. And it's not completely clear if having more methyl groups or less methyl groups in your diet is what's contributing to this, but what we do know for sure is that there's certain chemicals which are called phytochemicals. They're found in tea, polyphenols, which are found in berries, and curcumin, which is found in turmeric, and these are effective against cancer, and they act through epigenetic mechanisms, potentially to restore some of that methylation.
So you can change or control your epigenetics partially by diet, but have people developed any drugs to change your epigenetics, like in cancer? Are there drugs we can use to turn on those tumor suppressor genes and turn off the oncogenes?
I don't think there are drugs that can target that yet, because the issue with epigenetic marks is if you try and target something epigenetically, it could affect the rest of all your cells. What is being done is they're trying to develop drugs to target what are called these remodelers, which kind of remodel the genome and kind of move it around. And so they're trying to target those kind of drugs to prevent the cancer from spreading so quickly. But that's definitely an active area of research. So, and one other interesting thing about cancer is that cancer prefers anaerobic respiration, which is without oxygen. And the reason it wants this is because if cancer uses aerobic respiration, which is with oxygen, then that alerts the immune system that the cancer's kicking and fighting, and the cancer's trying to lay as low as possible. And what feeds cancer when it's trying to do respiration without oxygen is actually sugar. The way that cancer wants to grow is with sugar. So a lot of people are trying to deprive cancer of sugar by going on like a no sugar diet, or being very careful about what they're eating so that they aren't letting cancer go down this easy pathway and making cancer have to go down the hard pathway, which is alerting the immune cells. So epigenetics is a really cool field to study because in my mind it gives people hope. So a lot of times you hear people who get a 23andMe result back and they have both BRCA genes, which predispose you to breast cancer and ovarian cancer. And I feel like 10, 20 years ago, we would have been like, oh, you have this gene, you're going to get cancer. And now we're able to see a lot more of the environmental effects that play a role in genetics, and to realize, oh, we can try and eat healthier and exercise more, and that will likely not prevent us completely, but will likely lower our risks of getting certain diseases.
Yeah, and when you get these results back with 23andMe, these SNPs aren't 100% definite, you know, they're just predictors that you're likely or at risk. So, I mean, epigenetics is, you know, one of the ways that we can...
Yeah, and just like with my hair, not 23andMe saying I was only 6% likely. You can see I have red hair and I don't dye it. But ultimately with epigenetics, you do have to do a lot of research and reading to see what research has been done, because there definitely are people that are trying to market epigenetics as this cure-all thing. Eat this food and you will be cured, and make your body what it needs to be. On the flip side, there are people that are saying that epigenetics is not important to study because it's not changing anything, and people are getting way too hyped up about it for no reason. So with most things, epigenetics is kind of in the middle, where it can be very helpful, but we also have to be careful of what we believe. And ultimately, epigenetics is not going to affect everyone the same. Since epigenetics is based a lot on the environment, everyone's environment is different. So what may work for one person may not work for another person. So that's also important to acknowledge when you're talking about epigenetics.